outside. But I'm very happy that even during this COVID time, the University of Mysore Academic Staff College able to arrange a refresher course for you. Hope this would help you to achieve your objectives in your life. With this brief introduction, I thank again the Academic Staff College and the coordinator, and let me come back and share my presentation for today's session. I am going to talk about, hope you are able to see the presentation now.
Sir, make a double click to number 1 slide, sir, and it will be.
Are you able to see?
Yeah, full screen you make it, sir.
Yeah, it is. I made it full screen.
Oh, okay. Dear participants, is it okay? Yeah, no, okay.
Yes, sir. Okay, sir.
Yes, sir, it's okay, sir.
Yeah, please,
Thank you.
Please, sir.
Thank you. So the topic that I have selected, since it is an heterogeneous group, I don't want to dwell upon the very complicated aspects of some of the phytopathogens, disease-causing agents in plants. Especially, I have selected 2 important agents, Viroids and Phytoplasmas, because these 2 important agents are gaining importance, and we have been, in the last couple of years, a lot of research work has been done on these 2 agents. So therefore, there are several aspects have been introduced in PU board also, and even in undergraduate classes in first semester of MSc Botany, microbiology. These concepts have been recently introduced, Viroids and Phytoplasmas. I hope this presentation will help you to understand more, and you will be able to address certain issues related to the unusual phytopathogens, Viroids, and the Phytoplasmas. As you know, like human diseases, plants are also going to suffer from different types of disease-causing agents. We have bacteria, fungi, viruses, nematodes, protozoa, algae, and even to the fact that certain insects do cause damage, and the damage is a significant amount. Okay, so all these we study under a separate discipline called phytopathology or plant pathology. So before understanding the all other aspects of plant disease and phytopathology, let us try to understand what is a disease, what is a plant disease. Okay, so when a plant carries out its normal physiological functions, whether it may be absorption, translocation of water, photosynthesis, respiration, all other physiological process normally, and it flowers at a right time and produces flowering, and fruiting takes place in time. So we consider as a normal process. If there is any deviation in all these physiological process, starting from absorption of water to translocation, photosynthesis, respiration, any abnormal changes in the physiological process of the plant body, which ultimately disturb the normal activity of the plant organs and development. That's what we call it as plant disease. And it was British Mycological Society in 1950 gave one more definition: any harmful deviation from the normal functioning of the plant body, we call it as a disease. So it is also words put for human and animal diseases. So plant disease can be described like this: any changes, any abnormal changes in the plant body, we call it as a disease. There is a discipline in botany which takes care of plant diseases, and which completely focuses on study of plant diseases, causative agent, mechanism by which how disease development takes place, how to manage the disease. All these things we study in a separate discipline, a course like phytopathology, otherwise known as plant pathology. So plant pathology is the art of science of treating the sick plants. Why we need to treat the sick plant? Because it is very, very important. So the treating the sick plant helps us to improve the chances of survival of the crop plants. So you know that we are all dependent on crop plants for food. And if the healthy crop produces healthy food, we are healthy, and we will be very happy, isn't it? So as a science, plant pathology is a noble profession, noble goal of protecting the food that is available to humans and animals. And that is why it is very, very important. And at the same time, plant pathology or phytopathology, it is also an interesting and very challenging subject in the field of botany or the agriculture. The expert botanist or agriculture scientists who are trained in plant disease management practices are referred to as plant pathologists, otherwise known as plant doctors. So doctors, how they treat humans, and veterinary doctors treat veterinary, domestic animals. Similarly, we have a plant doctor concept, and plant pathologists are generally referred to as plant doctors. The task of plant doctor is to keep the plant healthy so that you will have healthy food, healthy, productivity, and we are all happy with eating the healthy food. Therefore, food is very, very important. The food security of the nation depends on the role of plant doctors, who always protect the croplands from ravages of disease-causing agents. So therefore, the science of plant pathology calls for the knowledge of pathogens. What are the different types of agents which cause diseases in plant, and how do they affect the growth? How do they affect yield and quality of the produce? Ultimately, we are all interested in the final product. Okay. So therefore, the plant doctors or the plant pathologists role is very, very important in protecting the crop plant from diseases, so that we will have, quality food. That is very, very important. There are different types of plant pathogens do cause diseases, which can be broadly classified into non-parasitic diseases and parasitic diseases. So non-parasitic diseases, nothing but abiotic agent, environmental certain conditions, abnormal environmental conditions. Okay. For example, excess of minerals. If you put too much minerals in the soil, there will be toxicity. If there is a deficiency of certain minerals, then there will be deficiency diseases. So therefore, mineral excess, mineral deficiency, toxicity of certain chemical, toxicity of certain chemical pollutant, extremes of environment, high temperature or low temperature, high oxygen, low oxygen, excessive water and moisture content, excessive rain, all these things, air pollutant, excessive pesticide, excessive fertilizers, all these comes under abiotic agent. They do cause diseases, and they do impair the normal physiological functions of the plant body. Apart from that, we have many living organisms, what we call biotic agent, which are otherwise called parasitic diseases. We do use the word parasitic diseases when a biotic agent cause diseases. What are these biotic agents? We have a bacteria, fungi, algae, protozoa, even higher plant, angiospermic parasites like Cuscuta, Dodder, Striga. Then we have nematodes, small eelworms, which are inhabitants of the soil. They do cause diseases. They eat away part of the root and makes the plant to suffer from absorption of minerals and water, and insects. Okay. And finally, we have phytoplasmas, viruses, and viroids. Very minute, very submicroscopic. We cannot say living organism, but they are called molecular parasite, do cause diseases. So all this comes under parasitic diseases. And put together, we, whenever these agents cause diseases, there will be physical manifestations of symptoms. Okay, disease symptoms or syndromes, they do appear on aerial part of the plant body. So you can see drying of leaves, yellowing of leaf spot diseases, in non-emergence of inflorescence and non-flowering. Okay, and finally no fruits. Without fruit setting, the tree will start dying. Various types of symptoms. We call it as, of course, pathologists do identify these symptoms, okay, based on the symptomatology. Apart from that, there are many root diseases, many pathogens. For example, I told you nematodes. They enter the root system and cause galls and various types of knots in the symptoms. Even in mulberry, even in carrot, they take away the edible part of the plant body. Okay, so which we are all dependent on such edible part, but ultimately they are all converted into unedible things, and therefore they do affect our food tray. Similarly, the yield, the flower size, fruit size, okay, all these things will be affected, and various types of disease symptoms appear, and thereby we can identify the different types of symptoms. Even so, I'm not going to share some of the disease symptoms. Maybe you are familiar with that, but I'm going to share some of the important images related to the phytoplasma and viroids later. Soon after harvest, the food that is stored even at that time during that post harvest period. So soon after harvest, we are going to dry the food, and until it reaches the consumer, we have to protect it. There will be loss during post harvest period. That loss is called post harvest disease problems. It is related to market pathology. Tons of loads of food grains will be lost even in India. Okay, and 10,000 crore worth food is wasted in India every year. You can imagine. If you protect that particular food, that food can be feed to millions who now go hungry. So 61,000 tons of food grain rotted way back in 1994. Okay, India wastes more fruits and vegetables in a year than are consumed in the United Kingdom. Look at the statistics. So this was the statistics that I have taken from the Outlook magazine. Food grains wasted in India could fed up to 117 million people in a year. So therefore so much of food is waste. Again, there are several different types of pathogens, biotic agent, abiotic agent cause this food loss. So therefore, here comes the role of pathologist, agriculture scientist. They need to save this food so that we can feed this food to people who are going hungry. So what Dr. M.S. Swaminathan said, Dr. M.S. Swaminathan, he was one of the pioneer of Green Revolution India, agriculture scientist. Okay, he said it is a shame if it cannot save its food grain for needy. Okay, the state should have been talking about food security. Providing food should be the priority. Instead, the government has chosen to focus on airports, Commonwealth, many things. But many people are dying. So therefore, it is also a shame. So therefore, the time has come. We need to give more food, protecting our food. It is not only important, we give importance for raising the plant. It is also important that we need to protect the produced food. Okay, very, very important. Why plant diseases are so important? In 1845, there was an Irish famine. I'm going to tell you something about, a brief history of Irish famine. In Ireland, it took place in 1845. Half of the Irish population died. Same number of people migrated to other parts of the world in search of food. Okay, so Irish famine caused by the Phytoplasma infestation, a fungus, and it completely destroyed the potato crop in Ireland, because Irish people mainly dependent on the potato crop, and therefore people were starved from 1845 to 48. Okay, so half of the population reduced in Ireland. So similarly, in 1880, a coffee rust disease, caused by a fungus, Hemilia vastrostic, a fungus which completely destroyed the coffee plantation in Sri Lanka. Now, if you visit Sri Lanka, all the coffee plantations have been replaced by rubber plantation. So that's why a plant disease, a simple plant disease, can completely wipe out a plant species, important crop species. That's why very, very important. In 1942, in Bengal famine, in our own country, India, 1 million, 2 million people died. Same number of people migrated from West Bengal to other parts of India in search of food. Okay, again, the fungus was Helminthosporium viriae, which completely destroyed the rice cultivation. So we could not harvest any rice in that area, 1942. Okay, and therefore there were many people died. Many people died from starvation. In 1970, southern corn leaf blight, that too in a developed country like USA, United States of America, a fungus like Helminthosporium maedis completely destroyed the corn plantation, corn cultivation, and they could not harvest a single grain. American economy was also affected. So therefore, plant disease can bring such a drastic changes and affect the social, political, and economic aspects of the country. That's why it is very, very important. So it has been conservatively estimated that, on a worldwide basis, 35 to 40% of the agriculture produce is being destroyed by the phytopathogens, plant pathogens. 30 to 45%, you can imagine, is a huge loss. Okay. In India, Niti Aayog in 2020 estimated that more than 10,000 crores of food is being lost. Okay. You can imagine. This is why, if a concerted efforts are not initiated to save these crop losses from disease causing agent, whatever we have achieved in the name of green revolution will be lost, and there will be a food problem everywhere. So we are all talking about food security, everything. Food is very, very important. We need to learn from sub-Saharan countries. We need to from other countries. We are in a country where throughout the year we can grow many different types of crops. So that's why we should feel proud that we are born in a country where a lot of agriculture crops can be grown and there is no dearth for food. But at the same time, we need to respect the nature. We need to give importance for protecting our food so that millions of people can be saved from starvation. Okay. When we talk about plant diseases, I'm not going to explain, some of the details of plant pathology, but I will take a small leads from my presentation and, coming to the topic. So there are 4 different types of diseases can be recognized. Okay. So today, whatever corona, COVID-19, it is a pandemic. This disease is there everywhere throughout the world. Okay. It occurs vast area, extensive, causing severe damage. Similarly, same pandemic disease was also occurred way back in 1845. So that Irish famine, I'm going to explain later. There are endemic diseases where they are restricted to one particular region of a country. Okay. So then epidemic disease, all of a sudden disease appears and disappears. So they appear and disappears. Okay. But if they appear in a destructive form. Such type of diseases called epidemic disease or epiphytic disease. There are other types of diseases called sporadic diseases here and there. Okay. So they do occur at irregular intervals and locations. And there comes once in a year or once in 2 years. Okay. But pandemic diseases are very, very important. When they come here, they do cause significant damage. So I'm going to explain one important disease that is called Irish famine. And Irish famine was, if you just type it in Google, you will understand what is Irish famine, and it is a disease related to late blight of potato. Okay. You know that Ireland and throughout the Europe, the potato was the #1 crop way back in 1845. And, the potato was actually a South American crop. It was introduced by Spanish to Europe. And it has become a #1 crop in throughout Europe. So also in Ireland. In early 1845, a late blight disease, okay, appeared in Ireland and devastated the vast areas of potato crop. So what has happened? Because Irish people dependent, they grow only one crop, that was the potato crop. So their main staple food was potato. So when the disease came in 1845, the late blight disease completely devastated not only in Ireland but also in other part of the Europe. Okay, within a span of 3 years, from 1845 to 48, this late blight disease was there and completely destroyed the potato cultivation in throughout the Europe. So what has happened? The fields which promised bumper harvest, all transformed into areas, all transformed into a rotten fields. Within a span of 15 days, the tubers all started decaying underground. They could not be able to take out the tubers. Okay. And what has happened? Because Irish people, they depended only the potato as a staple food. So they could not harvest. So hunger. So, everywhere there was a hunger. And some people started, eating the, spoiled potato tubers. They were all already infested with dysentery and typhoid fever, and thousands of people, 1.5 million Irish people died. 1.5 million Irish people died from hunger and the dysentery and typhoid fever. Okay. And it is due to the late blight disease. And it has so happened at the same time, all the Irish people, actually the landlords who were sitting in England, they given the land for, agriculture, for all these workers or the farmers were actually bonded laborers. The British, the landlords or royals, they brought a lot of pressure on the government in England, and they brought out an act called an Eviction Act, okay, where the tenants were thrown out of their homes in Ireland for not paying the rent, okay. So finally, help came from the United States of America and Canada, and millions of people migrated to, America and Canada. Oh, the 10% of the American, American population and Canadian population, they are actually the Irish people migrated way back in 1845 to 1950. Okay, they left the homeland and they became the, citizens of America and Canada. But at the same time, same number of people died. Okay, Ireland population reduced to 7 million from 14 million. You can imagine this statistic speaks about a severe rate of late blight disease, a single disease, how it can impact human population. So that is why plant diseases are very, very important. So from 1845 to 48, what has happened? The weather conditions were so congenial, okay, in Ireland and throughout Europe, a deadly mix of ignorance and politics responsible for the Irish famine. And, later, whatever happened, it was a history. Tenants were thrown out of their homes in Ireland for not paying the rent. It was illustrated, London News, 1848 published all this data. And Eviction Act was brought, and all the tenants were thrown out of their house, and homeless families had to take a shelter on the countryside, okay, countryside. And finally, they all migrated, help came from the United States of America and also Canada. Some people migrated. Many people died in the airport itself, seaport itself, okay. And there was a, monumental Irish famine memorial in Dublin, okay. It was, that speaks about the, the sad picture of late blight disease, a single disease, how it has affected the Ireland's demographic, political, and cultural landscape. That's why plant diseases are very, very important. So after the first emergence of late blight disease, it took another 11-10 years to investigate the causative agent. What was the causative? Why late blight disease, came to Ireland and Europe? Why? How how it could destroy such a vast area? So finally, in 1861, Anton de Bary, okay, he was a German botanist. He did a simple experiment and finally proved that a fungus was associated with a late blight disease of potato, and he named the causative agent, disease causing agent, as Phytophthora. Phyto means plant, phthora means destroyer. Phytophthora means plant destroyer, responsible for the disease late blight of potato, and he became father of plant pathology. A systematic discipline emerged in 1861. So that is what we call plant pathology. We, the plant pathologists, study this particular discipline. So credit goes to Anton de Bary for discovering the causative agent. Since then, the discipline of plant pathology is one of the finest subjects within the program botany. There are different types of plant diseases, okay. These pathogens, there are different types of pathogens, fungi, bacteria, viruses, they cause root diseases. They all damage the root system and bring about diseases. There are pathogens, disease-causing agent, which cause diseases on stem region. There are some leaf diseases, bud diseases, seedling diseases, flower diseases, fruit diseases. Some of them, they transmitted through seed-borne diseases. There are some of the pathogens which concentrate on the vascular tissues. Plant body. So in the plant, within the plant body, there is a water-conducting tissue. There is a tissue which translocates water and minerals from root system to aerial part of the plant body, okay. So that is called vasculature, okay. And similarly, there is another tissue called the phloem, which transports the ready-made food from leaves to the other part of the plant body. Vascular system consists of the water conducting tissue and food transporting tissue. So what are these, xylem and phloem? We call it as a vascular in a plant body consists of xylem and phloem, water conducting tissue, and food conducting tissue. Okay, so there are pathogens which colonize this vascular region, xylem and phloem, and cause diseases. Okay, that's why they are very, very important. Water conducting tissue in a plant body, we call it as a xylem cells. When these xylem cells are infected, okay, there will be a disease called Wilted disease. Though sufficient water is present in the soil, the plant body unable to pump the water to the aerial part of the plant body. So therefore, aerial part of the plant body suffer from lack of water. As a result, such plants will wilt and they soon die. So such diseases called vascular wilt diseases. Many fungi, many bacteria cause such diseases. Okay. Apart from this, there's a food conducting tissue in plant body. We call it as a phloem. Okay. So look at the phloem structure. Okay. This is a phloem structure. Phloem consists of a sieve tube. There will be a sieve tube and there is a companion cell, and surrounding these sieve tube and companion cell, there will be a phloem parenchyma cells will be there. There are some pathogens which colonize these phloem sieve tube elements. Only because the sieve tube element is the food conducting tissue, very, very important. And it is present in every part of the plant body from root tip to shoot tip. And there are pathogens which colonize this specialized region, phloem sieve tube. That is why these pathogens, I call it as very unusual pathogens. Normally, generally pathogen won't colonize this particular tissue. Only specialized pathogens like viruses, viroids, and phytoplasmas, colonize this phloem sieve and cause diseases in cropland. Both viroids and phytoplasma, in that way, they are very, very unusual. So with this background, let me come back to my topic. Okay, the viroids and phytoplasma. I have divided my presentation into two parts. The first part, I am going to tell about what are these viroids, and the second part of my presentation will be on phytoplasmas. So let us discuss one by one. As I mentioned in my first part of initial part of my presentation, potato is the staple vegetable food crop. Okay, we the botanist, agriculture scientists, they study all agriculture crops. 95% of our food comes from about 10 to 20 crops. 18,000 higher plants are there, around 18,000 higher plants, out of which 80 plants are very, very important from the point of agriculture. 80 different types of plants. Okay, because we are directly dependent on all such 80 plants. Out of 80 plants, 15 to 20 plants are very, very important because they are crop plants. Because we are mainly dependent, because our staple food comes from these 15 to 20 crops. And among these 15 to 20 crops, potato, Solanum tuberosum, is one of the important vegetable crop. Of course, it was originated in South America. It is a South American crop, and it was first domesticated in Peru. Okay, first domesticated in the region of Peru. And there are about 5,000 different types of potato varieties are there. So whatever we are eating, only one type of potato, you imagine 5,000 potato varieties are there worldwide. And actually Spanish, who introduced this potato crop to Europe. When they introduced to Europe, and weather conditions were so same, almost same, both in South America and also in other part of Europe. Okay, and the potato crop was adapted very well to temperate regions in Europe, and it became #1 crop. Okay, in the 16th century it was introduced, and since then this is a staple food crop, and it has spread to other part of the world, and it became one of the staple food crops in Europe and also rest of the world. So the importance of that. Okay, so way back in 1922 there was a disease appeared. Okay, in 1922, potato spindle tuber disease. Okay, the name itself tells you potato tubers become spindle-like. Look at the picture, you can understand. Instead of oval-shaped potato tubers, whenever there was a disease, okay, all tubers used to become very, narrow and spindle type, and the disease was affecting seriously all commercial varieties of potato in United States of America and also other parts of the world. So the disease appeared in 1922. Look at, until 1967, no attempts were made to identify the causative agent. What was the agent? Okay. So they could not be able to understand. People, believed that it must have been caused by a viruses. So people believed that, so the potato spindle tuber disease was actually caused by a viruses, and they were trying to isolate the viruses. Viruses are nothing but nucleic acid surrounded by a protein coat. Okay, like capsid. Okay, nucleic acids. Viruses are generally, there are three types of viruses: plant viruses, animal viruses, and also bacterial viruses. We call them as bacteriophage. Plant viruses generally made up of RNA, nucleic acid, normally RNA, single stranded RNA will be there, externally surrounded by a protein coat called capsid. Okay. When we say virus, nucleic acid will be there, surrounded by a protein coat. So therefore, scientists were trying to isolate a virus particle containing nucleic acid and also protein coat. Okay. But they failed. So several times, several experiments have been conducted, and they could not be able to isolate the viruses. They could not isolate, able to isolate the viruses. They repeated several times. Then came in 1971, there was a landmark publication by an American pathologist. Okay, American plant pathologist. His name was T.O. Daner. T.O. Daner, American. He was working in U.S. Department of Agriculture. How our IARI, Indian Agriculture Research Institute. Similarly, in America, we have a United States Department of Agriculture. He was a scientist. So he tried several times to isolate the causative agent, thinking that potato spindle tuber disease was caused by a viruses. But he could not succeed. He could not get the virus-like particles in the infected tissue. He repeated several times. On the other hand, he was able to isolate a small low molecular weight RNA without a protein coat. Okay. A small RNA, a piece of RNA, okay, it was very 10-fold smaller than the viral RNA, and without having a protein coat, externally covering structure called capsid. That capsid was completely absent. And that RNA was also able to replicate itself, without, host plant. So it could be able to replicate. These were the characteristic feature of the molecule, RNA molecule, a small naked RNA that he has isolated. And T.O. Daner used the term viroid. So T.O. Daner was the first person. He used the word viroid, and he proposed that potato spindle tuber disease caused by small infectious RNAs, not from viruses. Okay. He clearly he informed all the pathologists that viruses do not cause potato spindle tuber disease, and this potato spindle tuber disease is caused by a small RNA, a naked RNA. And he proved these agents were causative agents of potato spindle tuber agent consistently. Today, after his initial discovery in 1971, okay, there are more than 40 different types of small RNA variations have been isolated in economically important crop plants. Okay. And it was a Herculean task for T.O. Daner to convince the international plant pathologists, particularly virologists, that virus do not cause potato spindle tuber disease. It was in 1887, International Congress of Virology. At that time, there was an Andre Loff. He was one of the most respected virologists at that time. When T.O. Daner convinced him, he said that the time has come for viroids. They are currently regarded as the lowest step of biological scale molecular parasite at the frontiers of life. So in the International Congress of Virology, the president of the International Congress, they accepted the T.O. Daner contention that the potato spindle tuber disease was caused by a small RNA. Till that time, no one ever believed that they caused by a small RNA. And he used the word viroids. Then let us try to understand what are these viroids. So viroids are nothing but very small circular RNA capable of autonomous replication. Look at the word autonomous replication, and cause diseases in plants. Small RNA, circular RNA, naked RNA, able to cause diseases in plants, we can call them as viroids. They are 10-fold smaller than viruses. So viral RNA. Okay, in viruses also RNA will be there, but the RNA nucleotide size is very, very small. How small? Look at the size. Small size, just about 250 to 450, less than 500 nucleotide bases, adenine, guanine, cytosine, urea, something, 250 to 450 nucleotide bases able to cause a disease. And in that 250 to 450 nucleotide, there is no coding region which code for protein code. Okay, there is no coding region. How do they do? Some viroids, they use host encoded polymerase. How do they replicate? They use the host polymerase 2 enzyme and they replicate. There are other viroids, even they don't use host coded polymerase 2 enzyme. On the other hand, viroids have catalytic activity, ribozyme activity. These small RNA capable of replicating themselves. A RNA act like an enzyme. That is what we call ribozyme activity. And they are mechanically transmitted through seeds, pollens, and insects are going to play a very important role in spreading these RNAs, small RNAs, from infected plant to healthy plant. So the word classical viroids have been found, and these type of viroids are known to cause diseases only in plants, not in animals. And therefore, lot of research work has been done, and these small RNAs are known to resist exonucleases activity. Even if you treat with RNases, they are not able to degrade, and therefore that's why they are very, very important. So way back in 1990, Ricardo Flores, one of the finest authority on viroids, he gave a wonderful definition for viroid. Viroids are nothing but a small, single-stranded, circular, low molecular weight, naked RNAs. Naked RNA sequences ranges from 250 to 450 bases, able to cause diseases only in plant. This is one of the complete definition for viroids. Okay, so this is very, very important. So if you look at the genome size, as I mentioned, viroid genomes are extremely small and covalently circular. So the moment you isolate them, they will become circular because of the covalent similarity in nucleotide bases. They become circular, covalently circular. Genomes adopt a rod. Suppose if you isolate them, isolate from the plant body, they will look like a rod-like structure. Okay, genomes adopt a rod-like secondary structure in vitro. The rod-like structure of viroid contain 5 functional domains. If you analyze completely, there are 5 regions will be there in the viroids. So I will tell you what are these 5 structure of viroid, how a viroid looks like. As I mentioned, it is a circular, but the moment you isolate, it will become a rod shape. Okay, and in that rod shaped viroid, okay, RNA, there are 5 structural and functional domains have been included. So one central region is called central conserved region. In all the different types of viroid, the central conserved region, conserved central region, CCR region, is very, very common in all viruses. On either side of CCR, we have a pathogenicity region called P, pathogenicity domain. The other side, we have a variable domain. So we have CCR. On either side of CCR, we have pathogenicity domain and a variable domain. On either side of the CCR and pathogenicity and variable domain, we have left terminal repeat and right terminal repeat. So these are the main 5 domains of the virus. Look at this particular picture. As I mentioned, when you isolate, it becomes a rod shape. So we have a central conserved region. So this is very common in almost all viruses. CCR region, conserved central region. On either side of this, we have a pathogenicity region, and we have a variability region, and we have a right terminal end and a left terminal end. There are 2 types of, this is very common. This is a, structure of a viroid. So when we talk about, if you analyze further, there are 2 types of viroids can be recognized. One is the rod-shaped viroid. Look at this, rod shape. The other one somewhat looks like a hammerhead, just like a hammerhead. Okay, it takes a different secondary shape. Okay, like this. One is a rod shape. Normally they are circular inside the plant body. The moment you take out, they looks like a rod shape or hammerhead type. Okay, so this is again, and based on this, the viroids have been classified. So one particular family to which the viroids belongs includes Paspi Viroidae. The other one is Eviction Viroidae. Okay. So it is based on the, based on the structure of viroid. One category of viroid looks like rod shape. The other category, Eviction Viroidae, looks like hammer type. Okay. So this is I don't want to getting viroids can be classified into two types, PSTVD, okay, because the type example is potato spindle tuber viroid group. The other one is Eviction sun blotch viroid. Okay. So coming to important diseases caused by viroids. Potato spindle tuber disease, I'm going to explain a little bit about that later. There is one more disease called coconut kadang disease, citrus exocartis disease, chrysanthemum chlorotic mottle disease, chrysanthemum stunt viroid, avocado sun blotch disease, peach latent mosaic disease, hopstun disease, avocado sun blotch, grape, even in grape also. There are 40 different types of diseases are caused by these viroids. So how to detect them? So detection is a very, very, detection of virus, we know how difficult it is to detect, coronavirus. Okay. We are all, affected by coronavirus. Similarly, the viroids are much more smaller. So detection methods, why for many years, so, scientists struggle to detect. Now we have a powerful techniques are available. Morphological method, symptomatology. Now the expert pathologists are able to identify the, these viroids based on the symptoms. But some of the most of the symptoms are similar with viral diseases. And spectrophotometric RNA analysis. So UV spectrophotometer analysis will also give us an indication that whether normal RNA is there or an abnormal RNA is there. So that can be detected. Electron microscopic observation, we can detect. Bioassay on indicator plant. There are some susceptible indicator plants are there. If you swab the sap taken the infected plant, there will be appearance of symptoms. Such plants I will tell you later. Okay. There's another technique called, these are all molecular techniques, slot blot hybridization methods by using a specific probe. We know this, we have sequenced, all the sequence details are available. So we have probes are available. Using the probe, we can detect whether viroids is there in a particular plant or not. Then two-dimensional page analysis. I told you the viroids are different, distinctly different from mRNA, tRNA, and ribosomal RNA, okay, host RNA. And therefore, two-dimensional page on a polyacrylamide gel electrophoresis with a negative staining with silver nitrate will give you a picture whether viroid RNA is present or not. The other method is reverse transcription PCR method. As I mentioned, the viroids are RNA. RNA has to be converted into DNA, and DNA can be detected by using regular PCR method. Powerful tools, primers are available. We can do that. When you do the PCR method, the viroid gets amplified. That amplified viroid RNA can be sequenced. cDNA can be prepared and then sequenced, and sequence analysis will give you whether it is a host RNA or whether it is a pathogenic RNA. So these are the detection methods that can be used. There are bioassay methods. Indicator plants can be used. One is Cucumis sativus, cucumber plant, tomato plant, Lycopersicana esculent, Nicotiana benthamiana, tobacco plant. These are all highly sensitive to the viroid infection. Within 15 days, they will show you certain symptoms. That's why these plants are called indicator plant. So these can be used. Look at this particular picture. One is the control, uninoculated. The all other parts are inoculated with the viroid. Look at the reduction in size, reduction in leaf size. How viroids cause disease. Such plants will not, because these viroids will interfere with normal physiology of the plant. Flowering won't take place, fruit setting will not occur, and finally such plants will be greenish and one day they will die. Okay. So how do these viroids replicate? Viroids do not code for any protein, unlike viruses. In viruses, we have a region called, coding region, which normally code for certain one or two enzymes. But here, viroids do not code for any protein. Viroids use replication mechanism of the host plant, particularly host RNA polymerase II. Okay, the host cell enzyme is generally associated with the synthesis of messenger RNA. They make use of this polymerase, RNA polymerase 2, and synthesizes the viroid RNA and viroid RNA. So all viroids, okay, potato spindle tuber viroid group of RNA, they do this replication. There are other type of viroid, they are self-replicating. Viroid act like an enzyme. They are called ribozymes. They are called hammerhead type of viroids. They have a catalytic property, self-cleaving, self-ligation occurs, and they multiply inside the host plant. So viroids, people have given a model to explain how viroids replicate inside the plant body. Okay, the viroid, it is, as I mentioned, it is a small circular plus strand RNA. Plus strand RNA. Once it enters, I said, I told you it uses nucleus encoded polymerase enzyme, polymerase 2 enzyme. The nuclear encoded polymerase enzyme 2 synthesizes a new strand of RNA and the plus strand, and it has got a ribozyme activity, self-cleaving occurs, and then there is a RNA ligase enzyme which joins this linear RNA molecule and it becomes circularized minus strand. This minus strand in turn act like a template for the synthesis of plus strand RNA, right? And again, ribozyme activity, the self-cleaving activity takes place, it becomes linear. Again, RNA ligase enzyme joins the end-to-end linear end RNA and becomes circular, and plus strand viroid is synthesized. And these plus strand viroid RNA circulate within the phloem cell and they migrate, translocate from infected cell to normal cell and causing diseases in the entire plant body. Okay. Little is known about the mechanism by which viroids cause diseases in plant. How do they cause? What is the mechanism? Not many things have been revealed, but many serious malformation do occur. Cell wall malformation occur. Membranous bodies, they get distorted, disorganization, chlorotic disturbances in the cellular level, hormonal expression. Okay, there are many different essential proteins which are required for normal metabolism of the protein of the plant body get affected. Okay, whenever viroids are infected, such plants, there will be imbalance in the synthesis of normal proteins, and not only that, they are known to affect hormonal imbalance in the plant body, and certainly they are going to affect the normal biochemical pathways of the plant body. So that's why it is very, very important. And recent studies revealed that these small RNA bind to mRNA. Okay, bind to mRNA is always single stranded, and these viroid, whenever they enter, they bind with a mRNA, and there will be a double stranded RNA is formed. When there is a double-stranded RNA, there is a dicer molecule which cleave the double-stranded RNA. So in the plant or animal body, only single-stranded RNA exists, whether it is a tRNA, mRNA, or ribosomal RNA. They are all single-stranded. Whenever there is a double-stranded RNA, this double-stranded RNA will not exist. Okay. So therefore, the dicer molecule will cleave that, and as a